Hey guys, how you going? So welcome to chapter 13. In this chapter, we're going to actually move into the second area of study for this unit, which is looking at how we explain inheritance. Yeah, so this is kind of moving into looking at genetics, DNA, genes, and patterns of inheritance, and how all that kind of stuff works. Yeah, so that's going to be a bit of a switch over from looking at the kind of developmental biology which we were looking at before. Okay, so let's jump into it. So in this chapter, we're going to look at genomes, genes, and alleles. In this video in particular, I'm going to be talking mainly actually about DNA and giving you guys a bit of a history lesson on what DNA is. Okay, so genes, genomes, and alleles. Okay. Here's the key information about this chapter. You guys can have a look at it in your own time. But we're going to hop straight into it. So let's start with looking at DNA. Yeah, so DNA stands for deoxyribonucleic acid. Yeah, if I was to break it down, deoxy means to take the oxygen out of a ribose molecule, which is a type of sugar molecule. Nucleic acid being the type of molecule that DNA is a nucleic acid. Okay. So DNA is the genetic instructions for all living organisms. Hopefully by now you are familiar and well kind of versed in this particular molecule and all the remarkable things that it does, but I'm going to go over it briefly for you guys anyway. Okay, it codes for the production of different proteins. Here's the key thing. Yeah, DNA codes for protein. All the proteins in your body is created or assembled using a DNA base code as a bit of a blueprint as instructions for making those proteins you have. Okay. It is a sequence of different what we call bases, and it's represented by A, T, G, and C. And a nucleotide is what the DNA is made up of. Every DNA molecule is made up of a series of subunits called nucleotides. And every piece of RNA, which you're also going to learn about, also is made up of nucleotide as well. And the nucleotide has three main components, and they're really important because the structure of DNA also tells us its function. Okay, it contains a phosphate molecule, a sugar molecule, which is called a deoxyribose, and a nitrogen containing base. Yeah. And the only molecule that varies between each nucleotide is that nitrogen-containing base. The phosphate and the sugar molecules stay the same. Yeah. So if you were to think, imagine this was a strand or a single strand of DNA. Yeah. So there's your DNA, a single strand there. Yeah. Each one has this sugar molecule, and it's called deoxyribose. It's because it's a ribose sugar molecule, and it doesn't have an oxygen, which sugars normally have an oxygen. Yeah. Is connected to a phosphate molecule and a nitrogen-containing base. Here's your base there. This base is the T base, right? And what happens is the sugar molecule of one connects to the phosphate of the other, and then the next one contains a different base. Yeah, so you've got now these this backbone, so to say, or this spine of this really long molecule connected together by subunits, and only the T, A, and G are different between them. These parts here stay the same. Okay. Okay. Now there are four types of nucleotides. Understanding this is important. Adenine, represented by the letter A; thymine, represented by the letter T; guanine, G; and cytosine, C. So A, T, G, and C. And that's kind of what it looks like. Its molecular formula. So these are the chemical bonds that make up this. This sugar molecule. Here's the one that makes up the base, and here what makes up the phosphate. Yeah, so I won't go into too much details about the chemistry about it, but we use kind of really simple symbols to represent that, right? And here's what they actually look like. The four of them, you can see adenine and guanine is larger than thymine and cytosine, and there's a reason for that. Yeah, the adenine joins together and bonds with thymine, A to T, or uracil, which is what RNA uses instead of thymine, and guanine bonds with cytosine. So A to T and G to C, or if it's an RNA molecule, which is what bacteria use, it uses uracil instead of thymine. Okay. Now you may remember, if you are really good at remembering molecules and their chemical compositions, that adenine, the adenine nucleotide, looks strikingly similar to ATP, adenosine triphosphate. That's because they largely have the same molecule. You can see there's the deoxyribose part, and here is that extra oxygen or hydroxyl group. Yeah, and then you've got your base there, and then instead of a triple phosphate molecule, which is triphosphate over there in ATP, you've just got one single phosphate group there, which forms the backbone of the DNA. Right? That's just a bit of a side thing. Here's an atomic model for what DNA actually looks like, and there are the actual individual atoms. You can see there what represents the phosphorus, carbon, nitrogen, hydrogen, oxygen. These are the 5 atoms that make up DNA. Yeah, and you can see there that's sort of how they are arranged, as you can see. Okay, there's just another way of representing it. Here's the backbone. Yeah, this is the 5 end. 5 to 3, and here's the 3 to 5 end of the backbone. Okay, and the model for DNA. So DNA was originally developed by 2 scientists, James Watson and Francis Crick, in 1953. The structure of the DNA was this double helix. Each DNA contains 2 chains. Yeah, so one of the first things they found that was kind of stumping everyone was they originally thought that it was just a single helix strand, but as they sort of went through all those kind of experimentation and working with the model, they figured out that actually was a double helix chain, and this is really important. Yeah, the sugar and the phosphate groups were the backbone, and it was a nucleotide that was facing inwards on the inside of the double helix chain, which forms your base pairs. Yeah. Now this lining up of the base pair, A to T and G to C, occurs through what we call a weak hydrogen bond. Yeah. And that was where the information was in this DNA molecule. Yeah. The remarkable thing is that by knowing the structure of DNA, scientists could then figure out how DNA gave the instructions to life, because before that they knew that DNA was vital for life, but they couldn't figure out why and what was it about the DNA that allowed it to do that, and then they figured it out. Yeah. So by having a double helix structure with this kind of base pairing method, what you could then do is that DNA could actually be a template for its own replication. Yeah, if you know one side of the chain, you can actually figure out what the other side of the chain is, right? Because we know that A always pairs with T and G always pairs with C. So if you know that on this strand here is an A, G, A, C, then you know on the opposite end it has to be a T, C, T, C to match this base pair, which means DNA, by having only half, could form the template for its own replication. Yeah. In which this was particularly important because cells in organisms would always replicate, right? The base pairing can also form a code, yeah, by looking at the order in which the base pairs are arranged. It then acts as a code for the production of proteins, yeah. By TAC would code for a particular component of a protein, TGC another component, etc., etc. Yeah, and so you can figure out exactly how these instructions were given and how they were interpreted by cells in order to produce the proteins that they produce. Yeah, and the last thing they also realized was that by changing some of these nucleotides, you could then change the overall instructions, which would change the organism's proteins and create the mutation that you would observe in living things. Yeah, and so that remarkable kind of structure allowed us to explain what the function of DNA actually was, which is pretty amazing if you think about it, right? And so if you look at a cell, here's your cell. Inside the cell is a nucleus. Inside each nucleus are sets of chromosomes. The chromosomes uncoil into chromatids, which then uncoil again into a series of histones or nucleosomes, and then in its most fine form, in its most fine form, that gives the base pairing that you observe there wrapped around. So it's a very neatly coiled set of instructions within the cell, which is why in meiosis and mitosis this process was so delicate in making sure that these instructions were done and divided well inside the cell. Yeah. Okay, so what does it actually do? We're going to think about, okay, how does an organism get from the DNA base code to the living component of the cell that allows it to function? How does the DNA allow you to, for example, perform gas exchange in your respiratory system and exchange oxygen for carbon dioxide, right? How does it allow the muscles to function? How does it allow neurons to fire? What goes from one end to the other? Yeah. But before that, we're gonna stop it there for this video, and then in the next video, I'm going to give you guys a bit of a brief history on genes and where the genes come from, because that forms that kind of component of understanding how inheritance is so vital to organisms. Okay, so we'll stop it there. See you guys.